Okay, so shall we make a start? It's my pleasure to introduce Balazs Orzsik. Sorry. And Balazs is finally a PhD student working with Mara in CISC. And today he's going to be telling us about his work on partial volume correction to improve the performance of sodium MRI sequences. So over to you, Balazs.
Yeah, thank you. So yes, today I will talk a bit about the physiological roles of sodium, also why sodium is important from an imaging perspective, and also some of the complications with sodium images, including partial volume errors. I would like to acknowledge Iris, as we did most of this work together. Also Mara for helping to set up the sodium acquisitions, Guillaume for the reconstruction algorithms, and Neil for the discussions the ideas about this project. How can I move here? Okay, so as I said, first I will talk about the physiological importance of sodium. And sodium ion homeostasis is fundamental to all biology. Human metabolism is made of thousands of coupled chemical reactions, and these reactions are organized and coordinated to compartmentalization. So there are semi-permeable membranes separating compartments, and a transport mechanism had to evolve in order to acquire vital substrates such as glucose, release products, regulate metabolite concentrations, allow energy production, excretion of byproducts, and to regulate cell volume. Despite the variety of metabolites that crosses these membranes against their concentration gradients, a common mechanism evolved to power all of these transport processes, and that happens through the sodium-potassium ATPase. So this is a membrane-bound enzyme that uses the energy of hydrolysis of phosphate bond in order to push out 3 sodium ions against its concentration gradient and push 3 sodium ions out and push 2 potassium ions in against the concentration gradient. And that's why it has to use energy. And importantly, 70% of the neural ATP expenditure goes for powering this pump. So it really highlights the importance of this enzyme. And on here we can see that this is basically a summary of ionic homeostasis, and we see that the sodium-potassium pump is sort of a key. It doesn't just set the sodium-potassium concentrations, but also indirectly affects other processes such as calcium homeostasis. Also it indirectly affects cell volume, because water passes through aquaporin channels it tries to balance the ionic concentrations, and those ionic concentrations are set by the sodium-potassium ATPase. Another role of sodium is in countering the electrostatic forces on anionic macromolecules. So there are electric interactions between functional groups, and sodium plays an important role to balance these. And the outcome is that these interactions define the properties of these macromolecules, such as conformation, aggregation, solubility, and so on. So how can I move? So why are we interested in sodium imaging? The essential role of sodium in membrane transport that underlies so many of vital functions can be a site of dysfunction and disease. And the potential metabolic information that sodium can provide, that was the early driving force to develop sodium acquisition. So in the early days, they tried to evaluate sodium images similarly as proton images in a descriptive manner, so looking at the contrast between these different areas. However, this bears little clinically meaningful information. In order to get something out of sodium imaging, there has to be done some form of quantification. And basically through quantifications, we make parametric maps that allow us to model some kind of biochemical or structural process. So before I explain what I mean by that, I will show you what I mean by quantification. So here is an image of myself. These are the so-called sodium phantoms that we need to quantify the signal, to convert the signal into something something physiologically meaningful. So what we do is we place the phantoms around the head this is in the field of view, and then we create some kind of ROI. So here I took slices of one phantom and calculate the signal within the ROI, then using linear regression to find a linear model how to convert the arbitrary signal into total sodium concentration. And then once we have something like the TSC, or total sodium concentration, we can use that information to further model and get information from the images that we could not get from standard proton MRI. For example, here, this is a study where they compared a young subject with an elderly subject. They recorded they estimated the total sodium concentration. And from that, by using a two-compartment model, by assuming that the intra and extracellular concentrations are constant, they could model the cellular volume fraction. So the left image here is the TSC, same here, and this is the cellular volume fraction image. And what they found out in healthy elderly, we can see that the ventricles get bigger, there is more CSF infiltration, but the actual CVF values in the parenchyma did not change. And this brings us to a question that still remains to be answered. When does a subject with neurodegenerative disease deviate from the normal function? So when can we see this drop? And what is different as well in sodium MRI, that it doesn't serve as a biomarker. So it's not just a statistical correlation with disease, but it actually reflects some physiology in real time, and it can provide objective longitudinal monitoring of disease progression and also a response to treatment. And as I mentioned before, it can shine light on processes that are not apparent on standard proton MRI. So this was sort of the nice part of sodium MRI, and here we start to get into the problems. The sodium is the second most abundant MR active nucleus that have biological significance in the body. However, it has very low signal to noise ratio, and that can be attributed to its low concentration. So in the brain, the concentration of sodium is around 35 millimolars on average, while hydrogen is 80 molars actually. Yeah, 80, and then also it has 4 times lower intrinsic sensitivity to magnetic resonance than hydrogen. It is a quadrupolar nucleus, and that leads to further complications because this nucleus behaves differently when it is in an isotropic environment, such as the CSF, and has different energy level shifts when it is in a macromolecule because of the interactions with the anionic negative groups. So in tissue, sodium nucleus has a bi-exponential relaxation time, and the fast component of this is lost within few milliseconds. So historically, it was quite difficult to acquire reliable sodium images because almost all of the signal came from the CSF rather than from the tissue. However, improvements in like higher field strengths, like 3D scanners, and now they went even higher and improved gradient codes and electronics allowed us to make more reliable sodium measurements. Another thing that is very different in sodium MRI that is how the frequency, how the image is sampled. In normal proton MRI, it's sampled through in a Cartesian way. Here ultra short echo times and non-Cartesian projection sampling is necessary. So the image is sampled in a different way, and that leads to complications with the image resolution. And that's what I will move on to. So as I mentioned, the spatial resolution differs between projection spatial encoding and Cartesian approach of phase and frequency encoding, what is used in hydrogen MRI. In proton MRI, the voxel dimensions are the nominal resolution, while in projection imaging, the point spread function, which defines the intensities in the voxels, spread beyond the voxel dimension. So that basically means that if we have a voxel, gray matter voxel, it will have information and signal from the neighboring CSF and the neighboring white matter voxel as well. Also, it's important to highlight that in sodium MRI, the kind of resolution that we get, the nominal resolution that we get is 4 millimeter cubed. But this resolution, the actual underlying resolution is even worse. So there is signal sort of bleeding into the voxels, and there is mixing, and that creates significant partial volume errors. And there have been some methods which try to correct these errors. So one simple way that they did before is to co-register the sodium image to high resolution proton image, to like an MP region anatomical image, and reslice it and split it up into compartments. But this method doesn't take account partial the PSF, the point spread function, and which basically creates resolution mismatch. So when we think the co-registered image shows that there is a voxel which is 99% of gray matter, it actually will have signal from white matter and CSF as well. So what we try to do is to use partial volume correction methods to try to dissect out the so-called pure real signal. So the first method, actually, I will explain what partial volume in normal MRI terms are. So here you can even forget about the complications with point spread function. We can think about it. Let's think about it that that there is no influence of point spread function for the sake of it now. The signal, so because we are talking about quite a big voxels, 4 millimeter cube voxel, as a signal, a voxel, a sodium voxel will contain a fraction. So this is the fractional volume of gray matter. It will contain a fractional volume of white matter and CSF. All of these have different concentrations. And so basically the voxel will represent a mixture of this. Iris has developed a way to dissect out the pure information from a tissue by assuming that for any given voxel, this was developed for ASL. So here we are talking about perfusion rather than concentrations. But for any given voxel, the perfusion of a given tissue is constant over a region surrounding that voxel. So starting with this assumption, a set of linear equation can be set up and solved for the unknown pure tissue perfusion. So basically, this method calculates a signal or a value, calculates the perfusion for gray matter and white matter if it only contained gray and white matter. And basically we adapted this technique for sodium imaging, and we added another compartment, the CSF compartment, which in ASL is less important because the signal is zero in CSF. Also, we developed this other technique, which is based on Gaussians. So the Gaussian mixture is a function that is defined by a number of Gaussians. In this case, is defined by 3 Gaussians, one for gray matter, one for white matter, and one for CSF. And we try to get out the Gaussians from tissue where which we deemed as comprised entirely by that tissue. So I will show you on the next slide what I mean by it. And basically, each Gaussians are defined by a mean standard deviation and mixing probability factor, which basically which gives information about the weight, how much of that Gaussian is present in a voxel or in a kernel. And actually, so similarly as in the SLR method, maybe I forgot to highlight this, but for the SLR, we are talking about, we go to a voxel and we build a kernel around it, and we get the equations for the regression from the kernel, and then we move to a neighboring voxel and build another kernel. So in the Gaussian mixture model, we are doing the same thing, but we are solving for Gaussians using expectation maximization algorithm. It might sound complicated. That's why I put together this slide just to explain what really happens. So we start off with a sodium image, and we also collect high resolution anatomical image, T1 weighted image. We register the anatomical image to the sodium image, and we segment it. Then we threshold the segmented mask, and we build a Gaussian. And we're using the mean and the the mean and the standard deviation to initialize the Gaussian, and also for the mixing probability we use a prior as well, which is basically the mean of that tissue, like the mean of the tissue segmentation probability within that kernel. And then we run the expectation maximization, and it gives us the estimated mean and the estimated volume mixing. So what we aim to do here is to compare the efficacy of the SLR and the GMM method for extracting pure tissue signals from simulated data, and then we wanted to test how these perform on real data. So here I started. So here I want to show like some qualitative data, like what these algorithms actually produce. So in the top row, in the top row, this is the output of the SLR method. So these maps represent the pure tissue sodium concentration in gray matter, white matter, and CSF. So one way to qualitatively check whether the algorithm did what it meant to do is to look at how uniform the maps are, because in theory, if the sodium concentration in gray matter is constant, then we should achieve a uniform map. And just by qualitative visual inspection, we can see that the GMM in all of these seem more uniform than the SLR. And here we basically reconstructed the raw image into a corrected image. So it's basically taking these maps and multiplying by the fractional volumes. And here we can see that, like especially in GMM, that these blurs sort of disappear and we see a bit more structure. So we wanted to test it in a more empiric way. So we decided to simulate our sodium data. So here you can see the real sodium image, and we wanted to get something like this. And how we approached this is that we started with a high resolution T1 weighted image, which we co-registered to the T1 weighted image that we acquired with the sodium. So basically this image has the same spatial resolution as our sodium images. And we basically downsampled the high resolution anatomical image, and then we segmented it, and then we multiplied it by values. These are the sodium concentrations in gray, white, and CSF, and we got these values from sodium literature, where they estimated the concentrations in these different segments by a high magnetic field MRI, which can provide better spatial resolution. So that was done with 7 Tesla. Then we convoluted with the PSF, and finally we added Rayson noise. So I don't know like how good these are visually, but I think we got like quite close with it. So the next thing that we wanted to decide on is how much convolution we need to have comparable simulated and real data. So here I used QLIV, which is quality index based on local variation. And basically what this method does, it creates small windows, 4x4 voxels, and it slides through each slice and calculates a mean and standard deviation, how the data is spread in that window, and it compares a ground truth and a simulated or simulated image, and calculates an index ranging from 0 to 1, 1 meaning that there is perfect match, and 0 that they don't match at all. And then we basically tested the true image. Here means that it's without convolution, and then we just widened the PSF, and we found out that the best match was when we convoluted with the center of the PSF 3x3 voxels, like yeah. And then we tested the performance on these simulated images. So the first thing that is apparent is that, so the true image is without convolution. This is true plus Rayschen noise. The true that is apparent that even without PSF, there is a lot of partial voluming. Here we are looking at the gray matter content mixing with CSF, and we see that when the gray matter content is gray matter content is low, we have a lot of CSF signal flowing in. And even when we go to like sort of high tissue volume, tissue fractions, like between 60 and 90, it's still overestimated by 50%. And here we see that the SLR method basically solved the line here represents what the true starting concentration was. This is the ground truth, that the SLR method gets the ground truth in both in true and true plus noise images, while the GMM overestimates it, and it's 35% less accurate in the true images. The benefit that GMM gives comes in once we start to convolute the images, where basically there is mismatch between the underlying anatomical image and the sodium image. And here the GMM on average achieved 10% more accurate estimations. One thing that I want to highlight here is that both methods are independent of the mixing of tissues. So they achieve the same values, fairly similar values, almost the same values at each different tissue mixing. And this is one of the sort of face validity test for a partial volume correction algorithm, that it should achieve the same results at each tissue mixing. Also, another way to check it is that they are at high tissue fraction. So basically 90 and 100% gray matter, the values are converging. So we looked at white matter as well. In white matter is more sort of dense, and there is not much CSF and white matter volume, partial volume effects. And again, we see that the SLR gets the ground truth really well in the unconvoluted images, while GMM gets it right without noise, but then the noise brings in some errors, a bit of errors, let's put it that way. However, when we look at the pseudo images, so the convoluted images, the GMM either performs the same as SLR or outperforms it. So with the CSF, in general, the SLR algorithm outperformed GMM. Once we convolute the images, we see that both algorithms, like, they get very far from the ground truth. So once there is convolution, it's quite difficult to recover the signal. Also, for the GMM, it wasn't well. This is a proof of concept study, and more thinking, and definitely more like there is definitely a lot of room of improvements. And I think that's why we see the CSF effect, like the CSF is highly underestimated. Like, one reason might be, I go back to the initialization phase, that it seems like the CSF, even at 99%, has two compartments. And this has been reported before, that in sodium imaging, the values that you get out from the sulci, that it follows a different distribution than the values within the ventricles. So in the future, to improve the model, we could model CSF as two compartments, and hopefully we can get better results in this tissue. So finally, what we set out to do is to test these methods on real subject data. And here we collected sodium MRI images of 14 participants. And then we wanted to look at regions where we expect the most of partial voluming. So between 0 and 20, actually there are not that many voxels, because we collected data from healthy young participants. So it would make more sense to test these on elderly participants when there is a reduction or where there is increased CSF and reduced gray matter fraction. But we see the same sort of trend that that GM or the GMM outperforms SLR and in gray and white matter. However, in CSF, SLR estimates the values better than GMM. So these numbers here, so here I put the mean and standard deviation of sodium concentration between 0 and 80% gray matter, white matter, and CSF. And as I mentioned before, when I showed the simulated data, that from high field strength MRI at 7 Tesla, the estimated concentration of gray matter is around 40 millimolars, which we get with on average with the GMM technique, while SLR corrects less here. Also for the white matter, the reported values are around 30 millimolars, which and that's what GMM calculates as well. And yes, with the CSF, we see that we are really underestimating the sodium concentration. Also, another thing that I didn't put up here, both methods reduce the coefficient of variation and the spread of the data. So I will just summarize what I talked about here. we tested two partial volume correction methods. Each algorithm produced a relatively constant tissue concentration over the varying amounts of fractional volumes of different tissues. In general, GMM method yielded values that were closer to ground truth in simulated data, and also it produced values that are closer to the physiologically reported, to the values reported in literature. Both methods perform similarly on real and simulated data, which is good, I guess, that it shows that our simulated data was somewhat close to the real data. However, there is still room to improve in our way of simulating sodium data. And we see that the PSF is a major cause of partial voluming in sodium imaging, and it seems like the Gaussian mixture model can deal with the issues with PSF better than the SLR method, and that is likely to originate because SLR uses the underlying tissue maps, the co-registered tissue maps as weights in the regression, and if there is mismatch between the resolution, the values that it uses to calculate the pure tissue is off, basically. So in the future, what we are planning to do is to test how these methods perform in simulated activation and in simulated lesions to take it further. And I will finish with this meme to cheer people up after this quite dry presentation. And I'm happy to answer questions. Thank you.
Thank you very much, Balazs. That's excellent work. Does anybody have any questions?
I've got a question. Are you building a different Gaussian mixture model for each voxel? Or are you just having priors on the responsibilities for the voxels? So are you calculating a different distribution for gray, white, and CSF for each voxel?
No. So the priors that are used for initialization. So if I go back here, wait, where did I put this? So I'm getting the priors I'm getting the mean and the variance from the 99% mask. So I assume that is basically pure gray matter in some sense, but of course that will be like convolution is like, it's not completely pure because of the effect of the convolution. What we are changing voxel by voxel is the initialization with the mixtures, so the mixing probability factor, and that is calculated from, so basically the same kernel that is built around the signal is built around the segmentation mask, and from that a mean value of the probability of that fraction is used to initialize the Gaussian mixture model, if that makes sense.
Yeah, sort of.
I mean,
yeah, I think the way sometimes people use Gaussian mixture models is you might just have a different responsibility for each class for each voxel or pixel, which ends up being kind of similar to what you have here anyway.
I mean, this is like, just had this idea, like didn't know much about it, tried it, and I mean, it's still in very, like, this is sort of very, very simplified way of correction there. I'm pretty sure there are a million things to do better in this one. So this was more of like a proof of concept.
It's cool. It's interesting. It feels like you could put the point spread function in this without too much hassle as part of like a forward model of the data.
Yeah, I think they've done, like, in the literature, some people did something similar and used irises sort of method. Am I right, Iris?
Yes. The one there are two things here. One, Balazs is humble and says that this is a simple version, but we should say that almost the same version is actually used in PET imaging. So we're encouraged by that maybe we haven't oversimplified it too much. The other thing is, I'm not quite sure if convolving these with a PSF is the right way to go with sodium imaging, because we may have spatial variation of PSF. So I'm not quite sure that convolving these Gaussians is correct. That would be an oversimplification in and of itself, but we can discuss this more.
I mean, wouldn't we need to deconvolve the Gaussian? Or, okay, never mind.
Yeah, from the real data, yes, you're correct. Deconvolve the Gaussian, yeah.
Yeah. So what I was suggesting was that you have a forward model which uses convolution to explain the data. So you don't ever do deconvolution. You only do convolution.
Okay.
So you're trying to understand like the underlying data, and then you have this weird point spread function, which you can calculate, which you can estimate based on the way K-space is acquired, right?
Right.
And so then you have this forward model which goes from your probabilistic model of the amount of sodium at each pixel goes through to explain the data.
But how is this different from what Balazs did? So basically he simulated the images, convoluted them, and then matched it with the acquired image. How is what you're saying different?
Don't you like? But I think what Ira means is for like real data, to come back from.
How can you convolve real data that has already been convolved? Maybe that's what I'm missing. Real data is already being convolved from the scanner, right?
I don't think that becomes a problem though, because you're trying to estimate this latent unknown like level of sodium mixture at each voxel, and then you just put it through this convolution process, and then you say this, and you try and make that latent representation look as much like it after going through convolution, explain the data as well as possible. So you try and fit it. You don't ever kind of backwards transform your data directly and fit the model. You're only ever trying to explain the data you observe after it's had, because the data already has this convolution effect on it.
And what would be the mathematical step of the explanation? Like, what do you mean mathematically explain the data?
Oh, okay. So what you do at each, you'd have a model for each voxel, which you then propagate through convolution through the partial volume point spread function convolution, and then you try and maximize the probability of the data under that transformed model. So it'd be easier if I could draw a picture, I think.
Yeah, and also because maybe I'm missing something, because the data is already affected by the PV, so I'm not quite sure how you apply that. And even if it's raw data, I mean, definitely it'll have to be done in raw data, not in K-space, right?
Yeah. It should work. It should work either way, I think. But yeah, I think I'll need to draw a picture to explain it a bit better.
Yeah, I'm definitely missing how this can. I like the idea, but I'm missing how it can be done without deconvolution. But we're not doing this for this paper, right? That's not what you're suggesting.
I think I get what you mean. Like, I don't understand the maths, but it's more like, you know, there is the unconvoluted data.
No, but there is no unconvoluted.
No, but like, that's what you try to solve. Like, that's
Exactly.
what you're solving for. And you just take in account, like, you know, you could put in motion and other things as like a prior to dissect out. So it's like,
Right, but then you
Exactly.
you write that equation on the other side of that. So you write, yeah, we have to write
yeah,
it
so
down.
it's like here and then.
When you bring it on the other side of the equation, that's the equivalent of deconvolution.
So you don't have to solve it linearly. It's a non-linear system, so you can estimate it using sampling. So you never solve the deconvolution problem.
Oh, so I see what you mean. So you reiterate and then you do whatever gives you the least error between the two sides of your equation. Is that's what you mean?
That's exactly right. Yeah.
Oh,
So
I get it now. Yes. Okay. So, and why would that be better in the terms of, I see, because then you're, okay. Yeah, that'll be a clever way of doing it.
Because like they used the convolution in like previously, but I think what they did, they actually convoluted the tissue segmentation maps, and they basically used the same SLR concept that like over a kernel, so it's basically just smoothing out the segmentation map.
Yeah, I'm not quite sure that's done for the same purpose. I think that convolution is done for resolution matching between the high resolution of T1 weighted and the low.
But like, I mean, that's what you need to run SLR correctly. Like that's why if we go to simulated data, or at least that's my understanding, that, you know, like when it's true, when the matching of the resolution is perfect, because here it is perfect because we created the simulated data from the segmentation maps, we get perfect answers. But once we convolute the signal image or concentration image, and we keep the segmentation images the same, we can't estimate it properly. Like, that's how I understood the paper.
Well, there are two different. There are two. I mean, I think they both affect, but they're two different processes. So one is the resolution mismatch, independent of the PSF of sodium or ASL for that matter. You have a resolution mismatch between high resolution T1 when segmentation tissue, the PV maps are gotten.
I didn't understand that. Like, why is it independent? Like, isn't it like the, you know, like, is it like how I understand?
Even if you have perfect PSF, you have acquired an image with 4x4x4 in sodium. Yeah. No PSF, just 4x4. Then you have acquired an image with 1x1x1 in the high resolution space, from which you get the partial voluming. So that mismatch between the metabolic beta ASL or sodium and the T1 weighted will introduce errors in your partial volume correction algorithm. So that's what they do. What they do is they get, I know this in ASL, so I don't know in sodium. They get an ASL image, and then what they do is they keep smoothing the segmented images until it matches the ASL image, and then they use that to get the PV values. So that's very different. I mean, it still introduces PV, but it's very different from the effect of PSF on the actual sodium data that is independent of T1. You can get anatomical images, not necessarily from T1. You can get them from, and actually people are trying to figure out how you can get the PV not from high resolution T1. I know that Michael has done some work on this because of the circularity in the problem. But anyway, there are two independent things: the resolution matching with T1 weighted, PSF, and nominal resolution. There are three different sources of PVE. And we didn't do, I mean, we did resolution matching only for the simulated data, but not with the T1 weighted. Am I making sense? Maybe not. It's so hard to do this via Zoom. So
I have a question. So to what extent does the quality of your segmentation affect this? Because it seems to be a lot of what you're doing is relying on the segmentation, and you just mentioned you just do a segmentation, but there's many different ways to do segmentation, and it can vary in quality. Have you ever done a really gold standard like manual segmentation and compared that to an algorithmic segmentation? To what extent does that affect what you're doing?
I think for the GMM, how the GMM is run now, I don't think it would affect it by that much, because it doesn't actually take the, it takes just it takes like a smoothed version of the segmentation as an initializing parameter.
And do you run the segmentation on an already downsampled?
Yeah, we tried to do it the other way around, and it didn't make a difference.
Interesting.
So what I was thinking though, which is similar, well, it comes to your question, is that when we are defining pure tissues, rather than using the whole cortex and saying that everything that is according to the segmentation is 99 is pure, using ROIs and trying to make another definition what we mean by pure, that the neighboring voxels have to be pure 99 as well. Because of the convolution, we are getting a lot of bleed through of the signal into other voxels. So what I was thinking about is to make an ROI or make a definition and go to cerebellum, where the gray matter is more densely pegged, or not densely pegged, but there are more neighboring pure gray matter values, rather than being on the fairly close to the surface, where we know that there is a lot of CSF mixing. And even looking at the distribution, we see that there are some voxels that are almost 80 millimolar of sodium, while it should be 40. So there is definitely a lot of signal coming in. So yeah, I mean that would be an interesting way. I try to get the signal up for the CSF from the ventricles, but that causes another problem, because then the CSF is modeled very weirdly around the in the sagittal sinus or around the cortex, because there is not such pure CSF that would have a mean of 100 and a much smaller standard deviation. So I think what you're saying, it's a good idea to use to do this manual segmentation or some better definitions for these Gaussians. And what I mentioned as well, it would be worth to model the CSF as two different compartments.
Just to add a little bit to this, Chris, this is your segmentation question is actually very important, at least for the SLR method and for ASL. We did some checking and we published the paper, but it's really not thorough. And I had never even thought about manual segmentation. So there are different algorithms there that we tried, but I think it's very much based on segmentation. So the entire thing of partial volume correction relies on good PV maps, which we don't know that whether we have them or not, especially with all the downsampling and what have you. Coming back to what Balazs said, one thing that Balazs does not know, because he has not looked at old brains, is how extremely difficult it is in studies of Alzheimer's and dementia and aging populations to find voxels with 95% and up tissue content. So I think that's going to be the main challenge of GMM, because that's when you have the most partial voluming, right? Brain atrophy is one of the main sources of partial voluming. that's when GMM may actually fail. So I think if we find a way to get away from defining these Gaussians on voxels with 95% and up, I think that would be huge for GMM.
Yeah, but I mean, in theory, we could even use literature values, or just try to use some kind of proxies, you know, like I don't know, some estimates from like big data sets or whatever.
Yeah, but then
So an easier way of doing it would be to, instead of having defining these things based on pure tissue probabilities, is that your priors are instead on the responsibilities for each of these, for each voxel, saying whether the voxel, each individual voxel contains white, gray matter, or CSF. So you learn the parameters from the data, and you can use whatever you can have a threshold saying it has to be above 50% for this class for it to count for anything, but you just learn it from all the data using the soft probabilities of the segmentations. Because that's how unified segmentation and quite a few other things work.
Right. That's actually a very good idea.
Yeah.
That's an
like,
idea.
Do you have any idea to how to? Well, I guess with what you suggested, that like model where you take into account the convolution in the model, that would sort it out. But my question would be that how do you cope with the resolution mismatch, that like that underlying tissue segmentation map doesn't actually reflect what's in the voxel itself. Like, I mean, I'm not solving that problem here at all. But if we're going further, I think like that is one of the main problems, sort of.
So I guess if you went to the to this kind of model where you incorporated the convolution when you're trying to fit your GMM, then it kind of, it wouldn't, because you're this latent model that you're building of the amount of sodium at each in each voxel in the brain is kind of irrespective of the PSF. I guess there might be some issues to do with resolution, but then the propagated tissue segmentation should still be like, you don't need to worry about convolving them with the PSF or anything like that, because you're kind of, the model that you build is of the real brain, and then you try and explain the data which has this strange PSF on top of it.
Okay, fascinating discussion. I think we should probably end there. We're getting close to 2 o'clock. So thank you very much again, Balazs.
Thank you.
Excellent talk. And thank you, everyone. I will see everyone next week.
Cheers.
Bye-bye, everyone.